Hi class, in this recording we're going to start talking about our stomach. And our stomach is super interesting. It's a sac. It's just this big muscular sac, this big blob of muscle with a lot of empty space in it. And as we look at this muscular sac, it's located within our left abdominal pelvic cavity, directly inferior to the diaphragm. So our diaphragm is going to go right up here over the superficial margin of our stomach. And the primary purpose of our stomach is to store food. Because we have a stomach, we can sit down, eat a whole bunch of food, and then walk away while we process that food. If we did not have a stomach, we would have to be continually eating tons and tons and tons of little teeny tiny meals. Take a bite here, a bite there, all day long. And as we look at our stomach, when it's completely emptied out, it has an internal volume of about 50 milliliters, so it can shrivel up, become quite tiny. But it can expand to up to 1 to 1.5 liters, or up to 1,000 or 1,500 milliliters after a typical meal. So you can have your stomach expand. This internal volume can increase a couple orders of magnitude. And if you completely stuff yourself, you can fit up to a gallon of food into your stomach. No problem. That's pretty typical. And there, of course, there's those atypical people like competitive eaters that can eat 50 hot dogs in 3 minutes. That's a little abnormal. Now, as we look at our stomach, it has both chemical and mechanical digestion. There's going to be mechanical digestion as it physically squeezes the food, and there's chemical digestion as it mixes the food with acid and digestive enzymes. The mixture in our stomach is called chyme. So when somebody vomits, that vomit that comes out of your mouth has a technical name. That name is chyme. So we send a big wad of chewed up food down into the stomach, the bolus, and when that bolus mixes with digestive enzymes and stomach juices or gastric juices, we recategorize it as chyme. Now, within the stomach, since we have a lot of acid and digestive enzymes within our stomach, it's important that we have a mucosa membrane protecting our stomach. The mucosal membrane is a simple columnar epithelium mucosa. And that mucosa and the submucosa are going to be stretched smooth or stretched flat as we fill out our stomach. But as our stomach drains, the stomach will shrink in on itself, and that mucosa will form some folds. And we call those folds in our stomach, in the empty stomach, a gastric rugae. And as we eat food and expand our stomach, those gastric rugae will stretch back out again and go away temporarily. Since we depend on so much mixing within our stomach to mechanically digest our food, we have a third layer of smooth muscle tissue in the stomach. And this is a key distinction. Not every part, or most of our alimentary canal will only have 2 layers of smooth muscle within its muscularis, but the stomach has 3. We have a longitudinal layer that goes the length of the stomach. We have a circular layer that transects the stomach. And then we have the oblique layer that kind of goes in between. And those 3 layers serve to really, that third layer, that oblique layer, helps us to really churn and mix the food while we are in our stomach, while it's in our stomach. Now, as we look at our stomach's microanatomy, on the inside of our stomach we have these tiny little holes, little microscopic holes. These little holes are referred to as gastric pits. Gastric meaning stomach, pit referring to the hole. And these are little holes within the mucosa of the stomach. And lining these holes, we have a simple columnar epithelium that extends all the way down. And within that simple columnar, or within this gastric pit, we have some glands that open up at the bottom. So we have one gastric pit, and then at the bottom we can have multiple glands, or branch points at the bottom. And as we look at these glands, we can have a pyloric gland, or we can have a gastric gland. And these pyloric or gastric glands are going to help us make certain kinds of digestive secretions within our stomach. There's a cardiac gland that's present only in the superficial, or excuse me, the superior margin of our stomach, known as the cardiac region. And as we look at the pyloric glands versus gastric glands, these glands are all going to correspond to different regions of the stomach. So we have cardiac glands in the cardiac region, pyloric glands in the pyloric region, and gastric glands in the gastric region. And the glands within a gastric pit can branch out into multiple sections. Let's back up a slide here just so I can emphasize those regions of the stomach. So in terms of the regions, the cardiac region of the stomach is right here. The pyloric region of the stomach is right here. And then right here is the main body of the stomach where we have everything else, where we have the gastric glands. Now, as we look at a generic gland, because these pyloric versus gastric versus cardiac glands all kind of have a similar layout, and that there's going to be a lot of similar cell types between them. They are all going to have a mucus cell, and these mucus cells, generally speaking, are going to be towards the top and secrete mucus that goes out towards the lumen of the stomach. And they're also going to have parietal cells. Most of the parietal cells are typically going to be in the upper half of the gland. So generally speaking, we have parietal cells next. And those parietal cells are going to secrete hydrochloric acid, and that's a big one right there. This parietal cells secrete a lot of hydrochloric acid. They also are going to secrete some intrinsic factor and a hormone that helps us to feel hungry, known as ghrelin. And then we also have chief cells. The chief cells are very numerous, particularly in the gastric glands. So we have lots of chief cells within these gastric glands, and there's not really that many chief cells. They're pretty much absent from the pyloric and cardiac glands. And the chief cells are going to release a key enzyme known as pepsinogen. They are also going to secrete gastric lipase, but the one that I'm very focused on is that pepsinogen. So for parietal cells, I really want you to focus on how they secrete hydrochloric acid, and for chief cells, I really want you to focus on how they secrete pepsinogen. We also have G cells, also known as enteroendocrine cells. Entero meaning digestive tract, endocrine referring to the fact that they make a hormone that travels through the bloodstream. And the enteroendocrine cells tend to be concentrated on the inferior margin, or the deepest part of the pits. And they are going to secrete hormones and paracrine messengers that we use to help regulate our digestive processes. That hydrochloric acid produced by the chief cells is not going to be all at once. Instead, the chief cells do it one piece at a time. So we take a hydrogen ion, and that hydrogen ion will be dumped into the lumen of the stomach, and then the chief cell will take a chloride ion and dump that into the lumen of the stomach. And a hydrogen ion plus a chloride ion in an aqueous solution gives us hydrochloric acid. It's important that the chief cells do this one ion at a time, that cation and anion, because if they had lots of hydrogen and chloride together in their cells, that would cause the chief cells to have a very acidic environment intracellularly, which would be potentially lethal for those chief cells, excuse me, the parietal cells. I'm not sure, I had a brain hiccup right there. That would cause the parietal cells to have a very acidic environment intracellularly, which could be potentially lethal for those parietal cells. And as that hydrochloric acid leaves the parietal cell and goes into the gastric lumen, it's going to take pepsinogen and turn it into pepsin. And it'll also activate the lingual lipase as well. The hydrochloric acid also is going to catalyze acid hydrolysis so that it can break up the connective tissues and plant cell walls that we consume. So it helps to liquefy our food to make it into chyme. It helps to chemically digest our food. So this hydrochloric acid works two ways. It activates digestive enzymes in our stomach, and it directly dissolves the food in our stomach. Our hydrochloric acid is also going to cause iron ions to change oxidation state. We'll go from ferric iron, Fe positive 3, to a ferrous iron, Fe2 positive. And that's good. We want those ions to be in Fe2 positive state so that they're more easily absorbed in the digestive tract and used to make hemoglobin. We also use the hydrochloric acid to give us part of our innate immune system. That hydrochloric acid causes the stomach to be pretty dang acidic. It has a pH regularly of about 2 in our stomach. And a pH of 2 is going to kill most microbes that get down into it. So even if you eat a piece of moldy cheese, most of those mold microorganisms are going to be completely destroyed in your stomach by the hydrochloric acid. Unfortunately, the same is true of also any probiotics that you may take. So if you're somebody who likes to take a lot of probiotics to aid in digestion, that's good. It's definitely not bad. But most of the probiotics that you consume, most of those colony forming units that you consume are going to be killed by your hydrochloric acid. Pepsin is one that we need to camp on for a while. As we look at pepsinogen, pepsinogen is what is actually secreted by the chief cells within our gastric pit. And pepsinogen is a kind of zymogen. A zymogen, generally speaking, is an inactive protein or an inactive digestive enzyme. And it's important that it's inactive because if this digestive enzyme was active when it was inside of the cells, for example, if pepsinogen was actively digesting proteins inside of the chief cell, that would cause the cell to digest itself from the inside out. In other words, the cell would kill itself. So the chief cells make an almost activated, so it's an inactive digestive enzyme, and then that inactive digestive enzyme is excreted into the lumen of the stomach, and while it is in the lumen of the stomach, it is activated, so then it can start digesting proteins. So as we look at pepsinogen, it needs to be exposed to hydrochloric acid in the lumen of the stomach. And while it's exposed to hydrochloric acid, there is acid hydrolysis that cleaves off some peptides, and the leftover piece is a pepsin. And pepsin is an enzyme that does a really good job of taking large protein molecules and chopping them up into little shorter peptides, or oligomers. And this protein digestion is not going to be completed in the stomach. These little fragments of amino acids, these small chains of amino acids, are partially digested proteins. We're going to continue breaking them down into individual monomers, or individual amino acid residues, in the small intestine. So that's all we have for this recording. If you have any questions or comments, please feel free to post them on the discussion boards or to shoot me an email. And as always, happy studies.